Right. Thank you very much. This is an excellent turnout for Nik's presentation. So, for those of you who don't know Nik Murphy is a PhD student working with me, and Nik's been around for a good few years. And I think first and primarily you're not so much a PhD student as a skydiver. I think that's the main descriptor for you. And I keep seeing these wonderful pictures that you have of skydiving, and so you're probably fitting the PhD in as a bit of a hobby in and around skydiving, I would imagine. So Nik's done a tremendous amount of work over the past couple of years, and really focused in on taking a look at visual hallucinations from a very mechanistic perspective, taking a look at using transcranial magnetic stimulation, EEG, and other neurophysiological approaches. And I know the initial first couple of months were extremely hard trying to bring these very difficult pieces of equipment together, but you tackled that task really wonderfully. And you've then developed it further, taking a look at sort of the hallucinations within disease populations, specifically Parkinson's disease patients. And we're really looking forward to just hearing more of what you've been doing and sort of celebrating your PhD. Now, Nik's, we've had a bit of a discussion, and we've agreed that you're going to answer maybe one or two questions from the audience, if that's okay. So if anybody's got any questions, Nik's very happy to take them, and I'm just going to let him crack on with it. So thanks, Nik.
Okay.
Switch to line. Is that okay for everyone?
Yeah.
Right, well, thank you very much for the kind introduction. As John Paul mentioned, I've been doing this for about 3.5 years now, and it's quite nice to have it all sort of come to an end. The last time I stood here in front of an audience talking, I barely had any data and was fairly nervous about presenting what I did have. So now it's a completely different kettle of fish to get to the end and have a PhD's worth of data to present. So I'd like to thank you all very much for turning up, and hopefully I'll be able to convince you why it's important to be studying visual hallucinations in Parkinson's disease with dementia and what we can learn from this using electrophysiology. Before beginning, I'll just give a quick outline of the talk. So, because not everybody here is going to have quite the wealth of knowledge of visual hallucinations or indeed Lewy body dementias as maybe some other people in the audience, I'll provide some background literature to bring everybody up to speed. I'll then bleed this into the aims of the PhD project before describing the methods utilised across the entire study, and then a summary of the EEG investigations. Finally, I will draw upon the conclusions and where this research could be going in the future. So, Parkinson's disease or dementia belongs to a category of dementias known as Lewy body dementias. Now, these are characterised by the presence of alpha-synuclein and ubiquitin within the presynaptic part of the neuron. As the pathology spreads, there is widespread loss of neurotransmitters and a reduction in the grey and white matter across the neocortex and the subcortex. The phenotype of this is memory loss, poor executive functioning, an array of different visual perceptual difficulties, and eventually psychosis in the form of visual hallucinations and dementia delusions. So visual hallucinations, as we're probably all quite aware, are the experience of having seen something that, whilst it might appear very real to all of us, or to whoever's experiencing it, it's a completely illusory percept. Now, in Lewy body dementias, this is common in up to 65% of cases, and over the course of the disease, as the pathology spreads and cognitive decline worsens, these will vary in complexity. They might start off very often as simple hallucinations, such as the presence of geometric shapes or patterns, maybe sort of feeling that there's sort of a sensation of somebody might be stood in the corner of your eye just out of sight, as well as sort of shadows and likes across our visual field. And then as the pathology spreads, these become more complex. The individual might begin to see things such as people, animals, household objects, and they would eventually be expected to begin to lose insight into the reality of this, in which case they might attempt to interact with these, not really being aware that they're not real. So as these become more complex and some of the things that might be seeing become more distressful or more frightening, it can increase the level of distress for the patient, which in turn has an effect on the level of distress for the caregiver, and can often end up resulting in having to place the patient in a care home or nursing home of some form. So obviously there's quite a significant impact on quality of life here for both the patient and their carer. So it's quite important that in order to treat this symptom effectively and to improve the quality of life, that we're able to find some form of effectively targeting the hallucinations and reducing their expression. So to do this, it's probably first best to talk about visual processing in general before trying to find where we might be able to look for a mechanism, as it were. So visual information processing begins its life in the retina. Light hits the eye, it's processed by different cells within different levels of the retina and sent along the pre-striate pathways through the optic chiasm and the lateral geniculate nucleus, which we can see running through the centre of the brain down here, and is projected out onto the primary visual cortex. Information is then processed and begins to be unpackaged at the primary visual cortex before being passed further along the visual system into different regions of the immediate cortex, and then eventually into further distributed regions to try and extract more advanced information. So these are located in the parietal cortex, which is known as the dorsal stream, such as the dorsal fin on a dolphin or shark or fish, and this is responsible for extracting spatial information that might be used for something like navigation or for interacting with an object in the environment. Now, the ventral stream runs through the temporal cortex along the bottom and is primarily responsible for extracting feature-based semantic information that we might use for recognizing what something is and trying to make sense of the item that's presented to us in our visual field. So, as we can see, visual perception is a very complex topic. It's the product of this is an interaction between processing in different streams, and all weighted by executive functioning going on that's in our frontal cortices, which is all the while trying to manage the different aspects of processing so that we're able to make sense of the environment around us. These complex hallucinations appear as very familiar items, and they will often appear very integrated into the visual scene. And so clearly, whatever's generating this is interacting with this complex system in a very specific manner. So it now becomes a problem to try and determine exactly where the problem is coming from. So over the course of history, people have studied lots of different aspects of visual processing, and in turn found lots of isolated effects and aberrant levels of processing occurring as a result of the pathology. But obviously, with what we're seeing so far here, it's difficult to kind of think that maybe one thing might be causing the hallucinations over rather than interaction. So, if we have a look at this from the level of visual input, we see through the literature that there's a history of retinal problems in both idiopathic and Parkinson's with dementia, such as reductions in the level of dopamine within the retina and to the morphology and functionality of the photoreceptors at different levels of the retina. We can study this indirectly using a technique called visual evoked potentials, and indeed this has been used for quite a long time and had quite a rich history of results. This is the waveform that we see on the second image, and this is generated by presenting a checkerboard-like pattern to the individual, which appears to flash, and when this flashes, this changes how the light is focused on the retina and triggers this response, which we record from electrodes that are placed on the surface of the back of the brain over where the occipital cortex should be lying, which is the primary visual area of the brain. This generates a three-peaked waveform, which gives us information about the quality of the information that's going in the first place, which is the amplitude of the signal, so the height of these peaks, and then the time information as to how long it takes for these different processes to occur gives us an idea of how efficiently information is being transferred. Now, classically, Parkinson's has been associated with a slowing of information transfer to the primary visual cortex. However, this has recently come under debate, given that some people from this institute had found that there was a reduction, or there wasn't really much effect of the distribution of Lewy body pathology in the lateral geniculate nucleus related to hallucinations in this group. So if we to move on further into the visual system, we can have a look at bottom-up perceptual processing, so as the brain begins to package the information. And across the cortex, there's reductions in grey matter in the temporal, occipital, frontal and parietal cortices, as well as a reduction in the metabolism in the primary visual cortex. Further to this, there's been an association between the density of Lewy bodies in the temporal cortex and the occurrence of visual hallucinations. And these changes to physiology and the anatomy of the visual system are quite fitting with what we see in the neuropsychological testing, whereby patients with Parkinson's disease, dementia, and dementia Lewy bodies across this Lewy body spectrum are impaired on, amongst other things, the processing of spatial and semantic information. Finally, we can look at visual processing from the level of top-down control over how all of this information is unpackaged and bound together. And this is often done in context of goals and the immediate environment around us to try and best make sense of the information. And we can consider top-down processing in two ways, such as gating of the information and trying to allow information to flow sensibly into the visual cortex, as well as acting to scale the level of processing in these different regions that we've seen are already impaired by the Parkinson's pathology. So this has been broken down into three particular control networks, which allow for the orienting of the visual system towards salient features or important features of visual information, and then trying to iterate through our working memory or our long-term memories to try and find a potential match for this, all the while guided by a particular attention network known as the dorsal attention network, which maintains the contextual focus. In Parkinson's with dementia, the dorsal attention network is particularly affected by the distribution of pathology, meaning that these patients have a very difficult time with trying to maintain the contextual information, therefore over-relying on the other two aspects of information processing, which are orienting us to important features and going through our memories to try and find a particular match. So a good example of this is provided in the image here, whereby a participant or patient might be looking at a hose, and because they're unaware or they're unable to sort of keep in mind precisely the context that this might be sort of in the garden, say, that we're in, the things that become immediately apparent are the shape, the size, maybe the texture of this, and so it's not unlikely that they might misperceive this and end up thinking that it was in fact a snake that they're seeing. So all of this tells us that visual hallucinations and the visual system in Parkinson's is quite affected in a fairly distributed manner, and that it's clearly not the case that there's a single dysfunction is primarily responsible for the hallucinations. And this has been discussed in many theoretical models over the years. A good example of this being one called the Perception Attention Deficit Model by Colleton and colleagues. It was developed about 10 years ago to explain how visual information processing relies on the input of the information to access our memories of what this information, or access from memory what this information could be representing, and to come up with potential matches known as proto-objects, and then to use the contextual information and our focus of attention to direct us to the most appropriate match. Now the stars on here represent the levels at which there are impairment in our patient group, showing that it's likely that hallucinations are not maybe just input and not maybe just the bottom-up processing of information, but in fact rely upon the integration of this dysfunction to result in the illusory percepts that we're seeing. So what we've looked at primarily in the background so far is fairly static methods of understanding the visual system in this disease group. And these are great. They do tell us exactly where there are problems, and they can even give us details of the particular neuropathology down to the chemical level of what's imbalanced in this group and what could be related to the occurrence of visual hallucinations. However, it doesn't really give us a very dynamic idea of exactly how the neurons and their patterns of communication are affected by the spread of this pathology. Now, electroencephalography, or EEG for short, is a measure of taking time point by time point relative to an event, or just investing the activity of neurons as they're kind of just going along millisecond to millisecond. This gives us an idea of synaptic communication in a far more mechanistic sense. For example, the image that I have down here on the bottom is an example of the visual response in the solid line to a frequently presented stimulus. And in the dashed line, we see a peak at about 300 milliseconds, known as a P300 component, and this is in response to infrequent stimulus. So we can measure this from particular points on the scalp and have a look that over time, in response to a novel item being presented, particular regions or clusters of neurons are more synchronous than in response to a different item that's being presented. So we get an idea of the time frame of perceptual processing. And there is a background of use of this in both clinical and control studies for visual perception and attention. Now, what we sacrifice with doing the EEG is the spatial resolution of the data, which is obviously very important for knowing precisely where we're getting this effect. We can seek to try and improve this by using a technique called transcranial magnetic current stimulation, or TMS for short, which allows us to stimulate very specific clusters of neurons to have a look at how they react in general, to get a look at the idea of their physiology, or to have a look at their functional properties. For example, this image that we see on the top here is taken from an MRI study by Caparelli and colleagues, who wanted to have a look at the regions of neurons recruited in response to occipital cortex stimulation during active perception of illusory percepts of light, known as phosphenes, which are generated artificially by the strength of the stimulation, compared to when there was nothing going on here. So what we see on the top is a region of functionally connected sites that are activated as a result of this stimulation during the phosphene perception, and then we can have a look at how this differs in the bottom panel when nothing's being perceived. Now, we can combine this with EEG to help improve the spatial resolution of our recordings, but also to have get an idea of the functional properties of these neurons. We can have a look at how they're responding in time, and we can just have a look at the general physiology of these, or we're able to use the study of phosphenes, for example, to have a look at how the system modulates the activity of these neurons in response to active versus passive perception. So the aims of my PhD project were to investigate the electrophysiological patterns of synaptic activity and how this was related to visual processing in PED at 3 different levels. So I focused on visual input, bottom-up processing, and top-down processing. Further to this, I sought to establish how the activity is related to the experience of hallucinations in PDD using measures of the severity of experience provided by both the patient and by the carer. To achieve this, with the help of our local recruitment network, the Dendron, I'd tested 17 healthy controls and 26 Parkinson's dementia patients. These were matched on age with a mean of about 75 years old. Visual hallucination experience was measured using the neuropsychiatric inventory for the carer's point of view on this, taking the product of the frequency and severity items of this battery, and then using an equivalent version of this for the patient, the Northeast Visual Hallucinations Index, by taking the product of the frequency and duration information provided. All EEG recordings were done using a 128 channel cap that gives us quite a detailed coverage of different locations across the scalp, and TMS was applied using a figure of 8 transcranial magnetic current stimulation coil over the occipital cortex, as is demonstrated on my wonderful volunteer down at the bottom here. So the first study had a look at the response in terms of visual input of the retina to the primary visual cortex. And this was done using the visual evoked potential technique that was described in the background. To do this, I presented the individual with a chessboard-like pattern that appeared to flash or to change phase every 500 milliseconds. Now, there's a large purple dot in the centre of the screen that was provided as a fixation point. The participants were asked to, if they can, to keep their gaze on this just to prevent the eye from wandering, perhaps picking up in the recording any activity that's related to completely different visual processes. The response to this was recorded from 3 electrodes placed on the back of the EEG cap and averaged over the 3 to provide better resolution of this signal, and I'd measured the amplitude and latency of the N1, P1 and N2 components demonstrated on the visual evoked potential waveform down here. So the findings of this were that the N1 and P1 component amplitudes were significantly reduced in the Parkinson's group. And this wasn't unexpected, as the visual acuity was also significantly lowered in this group, and that has previously been associated, as well as other factors in the retina, with a reduction in the amplitude of these components in the VEP signal in multiple different groups. It's not just a specific factor for Parkinson's. However, there was no significant reduction in the amplitude of the N2 component. When taking a look at the latency, the N1 and the P1 component latencies were not significantly different between the group, based on the literature which has previously used these components as a marker of the point at which information first arrives at the primary visual cortex. This probably tells us that there's some level, or to some extent, the transmission of information from the retina to the brain is actually intact in these groups. However, there was a significant difference in the latency of the N2 component. This is often lesser studied of the 3 and might well be an effect of a reduction in the white matter of these neurons. It's been noted previously in the literature, affecting the efficiency of the transfer of information further into more advanced processing. So I decided then that I wanted to have a look at this, how this related to the severity of the hallucinations. And so I'd taken all of the measurements we had and correlated these with both the carer and the patient ratings of hallucination severity. And what came out of this was that the P1 latency was significantly negatively correlated with both ratings of hallucination severity. Whereby as the severity of the hallucinations, which is on the y-axis, became worse, the latency of the P1 response appears to shorten or to become healthier, as it were, more in line with what we might expect to see in young controls. Now, we don't have sort of the physical data to exactly go and prove exactly why this is happening, but it's possible, based on what we've seen from recent neuropathology, that this could be due to changes in GABAergic control of interneurons, the primary visual cortex, allowing for the action potentials that are generated here to summate much faster. Whether or not this happens just as a general effect of the pathology, or if this is a compensatory approach, knowing that the processing much further upstream is far more inefficient in this group, in this case it might be that the latency is shortened, allowing more information to be processed at a quicker pace in order to flood the system with information, in order to improve the likelihood of making an accurate match based on the objects that are generated from memory. However, this would require some further research, both longitudinally to have a look at how this change develops over time and what symptoms it emerges around, and also would be interesting to have a look at the neuropathology at the end stage. We do at the moment have a megapress MRI component of the study as a whole that will be looking at some GABA spectroscopy of the visual cortex, so it should be possible in the not too distant future to be able to get some confirmation of the conclusions made here. So the significance of this is that we file information to support the notion that visual hallucinations possibly require a prerequisite state of healthy transfer to the primary visual cortex in order to occur in the first place, and that we might be seeing some form of compensatory shift in bottom-up attention, allowing for better processing of information further upstream. However, this will require future work on a larger cohort of participants in order to be more firm in the conclusions. The second study that I conducted looked at bottom-up perceptual processing in the occipital, temporal and parietal cortices between our groups using stimulation with TMS at the occipital cortex. Now, to do this, I placed the occipital cortex in a rostrocaudal fashion, so we've got the windings pointing upwards and the handle out to the side over the occipital lobe, and stimulated with the aim of attempting to find a point at which phosphines were possible to elicit, which was a bit of a difficult task in some participants, as it's not necessarily the easiest of things here, so I will be describing more of the general response in this. But for the purpose of this, the image on the right hand side is demonstrating a sham trial whereby the windings of the coil are tilted away to test whether or not there are any false positive reports as the stimulation strength becomes weaker with the distance from the scalp. Therefore shouldn't be depolarizing the axons to quite the same extent. I'd applied 240 pulses with the TMS in these groups, and 15 shams were interspaced around this. Now, a large part of the problem with combining TMS and EEG is the artifact that we end up seeing in the data as a result of the interference with the magnetic pulse and the circuitry in the EEG cap, as well as the tendency for this to create more muscular activity going on at the scalp from, especially with higher strength stimulation, sort of twitches in the cranial muscles and in the muscles lying directly underneath where it's being stimulated. This creates a rather complex artifact that is still not precisely understood. However, there is a wealth of literature emerging on sort of efficient techniques to be able to remove this. So a large part of my PhD had actually included developing a technique from this literature that would allow for efficient and valid measurement of the response that's come out of this. This involved using a combination of signal processing techniques, independent components analysis and empirical mode decomposition to break down the signal into smaller elements, whereby we can have a look at the pieces that make this up and take out the irrelevant information that is not actually generated by the cortex. Now, from this I then measured the amplitude and latency of the response at the occipital, central, parietal and temporal cortices as demonstrated on the scalp map to the right. And as the combination of TMS and EEG is still fairly recent, it's not been attempted in too many studies to date, possibly because of problems with the artifact and that, and it has, to my knowledge of the literature, not previously been performed in a Lewy body population. Therefore, it's not precisely known exactly what's occurring to generate each of the components that we see in the waveform. So I decided that it would be sensible to split this up into two analyses. So first of all, I've taken a look at the amplitude and latency of these components at each region between the groups, and found that at large the response to stimulation in all of these regions was very similar between the groups, with no significant differences in component amplitude and latency at any of the regions of interest. However, what we can see are there are some qualitative differences in the waveform that we wouldn't pick up using a typical window-based method to have a look at the peaks. This is obviously fairly important, as this is representative of information that's being processed at a more intermittent stage before this has gone on to form whatever process is occurring at the peak of this. So to achieve this, I had taken a time point by time point analysis and had a look at the distributions of the amplitudes between the group at each millisecond in the waveform, and where there was a significant difference between the groups, I had looked to find at least 20 milliseconds worth of consecutive significant differences in order to improve the reliability of this, and to try and guarantee that this was actually a real significant effect and not just caused by an outlier in the distribution. And what we see is that at each of the regions there's at least one period in time which was significantly different. and that's represented by the green bars that we can see on the plot, whereas the yellow bars are representing ones that were maybe 18 or 19 milliseconds that didn't quite make the cut, but this might change if the sample size is increased in future analysis. So what this might be representing is that there are some kind of changes in the neurotransmitters or the efficiency of synaptic communication, creating a slowing of the waveform. However, it's not entirely obvious exactly what's creating this, so it will be a point for future work to use more advanced analyses of the signal to break it down into maybe its spectral components to have a look. But the distribution of these significant effects does kind of overlap with what we've seen previously from the anatomical studies in bottom-up processing, whereby there are reductions in the metabolism at the occipital cortex, which obviously would affect very early processing that's going on, as we're seeing in the top graph, and then the changes to the structure of the parietal and the temporal cortices obviously being involved with information processing that occurs at a much later stage due to being more advanced information that's extracted, and therefore it's unsurprising that we're seeing some kind of differences much later on in these waveforms. Now, the significance of this is that we're seeing some similarities in the overall response pattern at the peak level, which gives us an idea that obviously whatever processes this is representing, they're not lost entirely within these groups. It's just that whatever the pathology is doing to these regions is causing the processing of information in this fashion to become inefficient. This might be due to additional factors such as neurotransmitter imbalance or general inefficiencies in synaptic communication, and will be a point to look at in future research. The final study that I conducted was looking at the application of top-down control of visual attention at the primary visual cortex level. Now, the visual system is constantly bombarded with information. We get an awful lot of stuff constantly being thrown at us from all different angles. Not all of this is necessarily relevant to what we're trying to focus our attention on currently. And therefore we need some kind of way of gating what information is coming in and being able to say, well, this is something that we kind of need to know that it's there, but we don't need to know exactly what it is, but maybe what's at the centre of our vision is far more important. Now, the correlate of this in EEG is known as alpha rhythm. Now, this is a series of oscillations that occur within the frequency range of 8 Hz to 14 Hz traditionally. And this can be measured in two fashions, providing us with information about the peak frequency, which is a measure of communication presumably between the cortex and its generating source, which is believed to be in the thalamus. And then we also get an idea of the strength of this, of the strength of inhibition that's being placed over these neurons, which is measured in terms of the power, which, as we can see on this image on the left, is to do with the amplitude of the response that we're getting. So a quick kind of overview of how alpha works in this sense. It acts to try and dynamically scale the level of inhibition that's occurring at the primary visual cortex neurons, so that we're able to maintain a focus of attention and to be able to detect relevant information. When the amplitude of the alpha is large, this represents a trend towards more maximal state of inhibition, and therefore visual detection is much lower, as we can see on the right of this image that's also to the right hand top corner of the screen. And then as the amplitude of the alpha power is reduced, there is less inhibition over the visual cortex neurons. They become more excitable and are more prone to being able to process the information, and therefore it becomes easier to try and detect a stimulus that might be being presented to this. Traditionally, in Parkinson's disease dementia, the power of the alpha response is reduced as a result of pathology, although this can be improved through the application of cholinesterase inhibitors. And we've also previously seen that there's a slowing of the peak frequency at which this is occurring into a range known as the theta range, although it's still not entirely sure as to exactly why this is occurring, but it previously hasn't been studied what the relation of these might be to the generation of hallucinations. So the findings of this that were found that, like in previous studies, was a slowing of the peak frequency of the alpha response in the Parkinson's disease dementia group, showing that there's some overlap in the higher frequencies, although the majority of participants tended to have this shift into a slower waveform, about 6 Hz, outside of the traditional alpha range. However, this was unrelated to the experience of visual hallucinations. Then, prior to having a look at the power of the response, I'd had a look at what effects of medication there were on this and found that there was a positive effect of cholinesterase inhibitors, which act to increase the power in those taking them, whereas those not taking them had a reduction in or had a lower alpha power than those taking them. So then when having a look at the power, what we see is that there's a similar level of alpha power in the Parkinson's group and the control group when the eyes are closed. However, when the eyes are opening, there's still the typical response of seeing a reduction in the alpha power representing a transition away from a state of inhibition. However, this is not quite as effective in the PDD group. However, what we might be seeing in terms of distribution here could be being upped by those taking cholinesterase inhibitors. Obviously, it's unethical to be able to ask our participants not to take these, and so we take account of the medications that they're on. But there was no relationship either way between the alpha power in any of these conditions and the experience of visual hallucinations. So significance of this is that perhaps the PD pathology is affecting the ability of this system to manage efficiently the control of visual attention and the overall neuron excitability. In those certainly not taking the cholinesterase inhibitors, there's certainly a state of more or less vigilant gating over information that's coming in. But what we might be seeing here is that whilst this effect happens generally in this disease group, it's not necessarily a central mechanism for generating hallucinations, but it may well act to predispose these participants to the experience of it by allowing more information to flow in, or to have less efficient control of the information that's going in the first place, which might then seek to confuse the processing of information that's occurring further down the line. So to conclude, my series of studies that I've conducted over the course of this PhD has provided some level of electrophysiological support for this integrative model of hallucinations described in the background, whereby we're seeing that there are some level of dysfunction in terms of both input, bottom-up perceptual processing, and the top-down control of the visual cortex neurons allowing this information to come in the first place. What we might be seeing is that the combination of this creates a state whereby it's now prerequisite for hallucinations, in which case there's less efficient processing going on, so it's more likely that hallucinations might occur, but it doesn't give us a precise idea of the generating mechanism that triggers the hallucination that we see. This will require longitudinal research as well as combination with multiple modalities of different research to arrive at a more solid conclusion. But certainly it does give us hope that it's possible to start understanding how, at least at this stage, we might better be able to treat the patients that are experiencing hallucinations based on this information. And certainly as more information comes out over time, we'll get a more solid idea of exactly what's going on. Now, I would like to thank quite an extensive list of people who've been part of this since day one. Obviously couldn't do this without the support of my supervisory teams, Dr. John Paul Taylor, Dr. Sora Graziadio and Professor Lynn Rochester. And then of course there's all the administrative staff, our neuropsychologists and our recruitment staff who've been a huge hand in being able to put this study together. So that's Alison Killen, Barbara Wilson, Heather Dowie and all of the staff involved in the Dendron team. I'd also like to offer my thanks to my panel of advisors, Professor Ian McKeith and Dr. Hamish McAllister-Williams, for their guidance over the years in our meetings, sort of helping to make sure the project stayed on track. I'd also like to offer my thanks to my friends and colleagues, Dr. Luis Pirata Rodriguez, Dr. Leo Tomasevic and Dr. Mark Baker, for their help on developing the technical knowledge required to put together the analyses and the design of this experiment. Without them, I would have been struggling coming from a psychology background to get so technically proficient to be able to perform the research that's been done. I'd also like to thank our Newcastle MR Centre radiographers for always greeting our participants and for making sure that when they go in for their scans and that they're comfortable and that they understand what's going on, they have a good time and come out and have actually enjoyed participating in this part of the research. And I'd like to offer my thanks as well to the carrier reception, admin and the cleaning staff, as well, as these people often don't get enough thanks and they create a fantastic environment for us to bring participants in for research. Without them this couldn't happen. They wouldn't want to come back and do more studies if they weren't as given as friendly a welcome as they are by the staff we have here. Obviously, it's worth to thank the volunteers and their families. This isn't possible without them, as they're the people that make the research happen and give us the data to be able to make these conclusions and to help others. And I'd like to offer my thanks as well to my family and friends who've been with me no matter where they joined the journey. You've supported me, encouraged me, and more importantly put up with me for God knows how long now. And then as well, thanks go to all the members, past, present, collaborating, affiliated, and of course future members of the Lewy Body Lab, and my previous academic mentors from both the University of York and Bangor University, who have helped guide me from day one of university right the way through to being able to reach the level that I have. That's all, so thank you. I'll take a few questions if anyone's got any. Thank you very much for your time.
Excellent talk, Nick.
Thank you.
So, any questions from the audience? We can take two. Ian?
So yeah, first class talk, I enjoyed it very much, and so congratulations on taking on a really complex area.
Thank you.
And explaining it to us. So a very simple clinical observation would be that people with Lewy body dementias seem to hallucinate most when their attention is diminished. So we used to bring people in and sit them in a kind of darkened room, and before they fall asleep fairly quickly, that's when they would hallucinate. So I was a bit surprised that in the final experiments that the eyes closed paradigm didn't really seem to show any great change. Did you think about other sort of manipulations of attention that you might have done with the patients to try and force them to hallucinate and sort of push changes in the system?
Okay, well, to be honest, I was as surprised as you are over this, as in the literature there is a rich history of participants having a lower alpha power, and this is often done with eyes closed as it's it's easier to determine the alpha peak, obviously with the much diminished response during eyes opening. And therefore I'm guessing a large amount of the response that we're seeing, obviously this is, the plot that we have is sort of from the mean group data, and so therefore there is going to be some effect of how the cholinesterase, and those taking the cholinesterase inhibitors are skewing the distribution slightly. As for other manipulations to attention, there certainly would be other ways of being able to do this. Like you said, it would be very interesting to sit these people in a darkened room and to just let them kind of relax and see what happens. We did, during the TMS, actually have a few people reported visual hallucination like phosphates. This is done with eyes closed, obviously. It's quite a long period of time, and quite often they can get a bit drowsy in that through the session. So I'd actually taken a look at these visual hallucination like phosphates in the 3 participants that had them, and tried to find if there was any kind of correlate to this in the clinical data that we've got and our visual perceptual scores and that as well, and found that there's sort of no general differences that are predisposing them to experience these. So whether this is just a result of maybe a lapse of attention through being sat down for so long is unclear, but certainly worth to test further. As for sort of direct ways of manipulating attention, I would have to sort of sit down and have a think about that and to really scrutinise what's been done previously in the literature to come up with something that's far more direct examination of this. As one person resting is very different from another person resting. It's very uncontrolled, unlike, say, an event-related study.
Thanks.
No, but thank you very much for the question. It's certainly something interesting to think about for the future.
I'll take one more question. You might get away with just one question.
Why not deal with patients? could you not include DLB previously?
We had originally, when we were designing the study, sought to include DLB, and certainly at the start of the recruitment phase of the study, we're still hoping that we would be able to. But to have, because originally this has been taking a far more dimensional approach looking at the hallucinations, because we weren't able to achieve as many non-hallucinators as we would have liked, and the original design of the study was to compare controls non-hallucinating and hallucinating. And with DLBs, it's more likely that they're going to be diagnosed based on the hallucinations, and therefore to try and acquire any with non-hallucinations was going to be quite a significant task. So by having the PDD, we get that level of flexibility where even though we've had to take, or I've had to take a look at the data in such the approach that I have, we've still got this cohort that have got some level of either minor or no hallucinations.
Great. Thank you very much, Nick.